Hello and welcome back. Today we are going to be covering the last section of this course, Learning and Memory. And before we begin, I wanted to share this beautiful image with you guys. This is known as the rainbow technique, and right now we are looking at the individual neurons within the dentate gyrus. So this is a layer of cells within the hippocampus. This is where neurogenesis typically occurs. Even in the adult brain, and we're going to talk more about neurogenesis in the dentate gyrus today. Here's a brief outline of today's lecture. First, we're going to discuss categorizing learning and memory. I'm going to talk about two key subtypes of memory known as declarative and non-declarative memory. We're going to discuss two types of learning, including non-associative versus associative. And we're also going to talk about learning in a very interesting species called the Aplysia californica. This is a sea slug that has become famous in neuroscience. We're also going to discuss declarative memory, such as brain areas involved in declarative memory, the hippocampus, and how this involves spatial memory processing. Finally, we'll end today's lecture by talking about different behavioral paradigms that are utilized in animal research to study memory. How are learning and memory different? Well, learning is the acquisition of new knowledge or skills, whereas memory is the retention of learned information. And if you remember, I previously noted that there are two subtypes of memory, including declarative and non-declarative. Later on, we're going to talk about the different brain areas that are largely affected by these different types of memory formation. We're going to talk about semantic memory, which is the memory of facts, such as geography. We're going to talk about episodic memory, which is the formation of memories like important events in your life, like your wedding. And we're also going to talk about different types of non-declarative memory, including procedural memory, skeletal musculature memory, and emotional responses. First, we're going to focus on non-declarative memory, which is also known as implicit memory. As I previously mentioned, there are three key subtypes of non-declarative memories, including procedural, skeletal musculature, and emotional responses. First, we're going to cover procedural memory, which is essentially our skills and habits like driving and riding a bike. These types of memories are largely regulated by the basal ganglia and the striatum. Skeletal musculature is largely regulated by the cerebellum, whereas emotional responses are largely going to be activating the amygdala. The striatum plays an important role in procedural memories, as I mentioned before, like driving or biking. The striatum, if you remember, is made up of both the caudate nucleus and the putamen, which make up the basal ganglia along with the substantia nigra. Remember, there are dopaminergic neurons that live within these key areas of the basal ganglia, and thus dopamine plays an important role in the formation of procedural memories. In order to better understand the procedural form of learning, I need to discuss non-associative learning. So, non-associative learning is a behavioral response to a single type of stimulus over time. And a perfect example of this is habituation, in which a person learns to ignore meaningless stimuli. For example, let's say you hear a car horn outside of your bedroom window. When you initially hear the car horn, it's going to evoke an increased response. However, over time, as the alarm continually rings, you're going to have a decreased response, and this is known as habituation. There's also another form of non-associative learning called sensitization. This is learning that intensifies the response to a stimuli. So, for example, stimulus could increase sensitization or a response to a particular stimuli. Eric Kandel is a renowned neuroscientist and leader in our field. He's known for his research on learning and memory in a model called the Aplysia californica. This is a sea slug, and it seems interesting that he would do the majority of his research in sea slugs, but we're going to utilize them in many of our examples today and why they have the perfect system to study neuroscience. There are many advantages of using a simple nervous system to study learning and memory, and a key one being that the aplasia californica have extremely large nerve cells that can be recorded. And this allowed Eric Kandel and his research teams to study learning and memory in the aplasia californica. Before we discuss learning and memory in the aplasia californica, I just want to talk about their sensory and motor neuron communication. Essentially, we're going to walk through the simple reflex in this sea slug. Interestingly, scientists have found that if you push on the siphon located right here, siphon skin is innervated with sensory neurons, similar to a human. Sensory neurons then synapse onto motor neurons, and motor neurons synapse onto the gill muscles, inducing a simple withdrawal reflex. This contraction of muscles from different stimuli on the siphon has allowed neuroscientists to study learning and memory. Measuring the simple reflex, such as a gill withdrawal in the aplasia californica, has been instrumental in the field of neuroscience. For example, researchers have found that if you stimulate the siphon of this sea slug, they are going to have this response, the gill withdrawal, and this allows us to form different forms of learning, such as non-associative learning, like habituation and sensitization. So let's walk through this example together. Scientists have found that if you stimulate the siphon with a water jet, this induces a gill withdrawal reflex. The water jet is going to induce action potentials in these sensory neurons shown here in pink. Action potentials in the sensory neurons are going to induce communication between the sensory neurons and the postsynaptic motor neurons. Neurotransmitters are released from the presynaptic neurons onto the postsynaptic motor neurons. Hopefully this is ringing a bell as we've talked about synaptic communication in our previous lectures. And remember that interneurons shown here in green are neurons that help facilitate communication between sensory and motor neurons. An interneuron is simply a neuron that communicates with other neurons. Ultimately, this release of neurotransmitters on these postsynaptic motor neurons shown here in purple generates excitatory postsynaptic potentials, or EPSPs, in the motor neurons. Although we've already covered excitatory postsynaptic potentials, or EPSPs, in this class, I'm going to briefly review it. So here we can see that there's a presynaptic neuron that's communicating with this postsynaptic neuron below it. We know that an action potential is arriving in the presynaptic terminal, and this induces the release of neurotransmitters. If you remember, this is exocytosis, when calcium comes into the presynaptic cell and induces this release of neurotransmitter containing synaptic vesicles. Neurotransmitters are then going to be released onto the postsynaptic neuron. During this time, sodium can enter into the postsynaptic cell, as shown here. Remember that sodium is going to come through these transmitter-gated ion channels that are permeable to sodium. This influx of sodium into the postsynaptic cell is going to induce depolarization due to this positive charge. Thus, this resulting change in membrane potential is known as an EPSP, excitatory postsynaptic potential. And if you remember, we've talked about the summation of multiple EPSPs occurring simultaneously that can potentially fire another action potential if a stimulus is large enough. This phenomenon can be recorded by a microelectrode, and this is exactly what Eric Kandel did in his research. He could insert these microelectrodes into the large nerve cells of the aplasia californica to measure EPSPs or IPSPs. And remember, when this membrane potential becomes more positive, this is known as depolarization. Now that we understand this simple reflex, known as the gill withdrawal in the aplasia californica, we're going to talk about a form of non-associative learning called habituation. Interestingly, Eric Kandel and his research team was able to study this form of learning by stimulating the siphon of these sea slugs with a water jet. If the siphon is repeatedly stimulated with a water jet, we're going to see that there is habituation that takes place. Repeated stimulation actually reduces excitatory postsynaptic potentials, EPSPs, as shown here in this image. Ultimately, the excitatory postsynaptic potential in the postsynaptic motor neuron gets smaller and smaller until it eventually habituates to the water jet. It is no longer surprised that it's receiving this stimulation. Thus, there is going to be reduced response of this simple reflex. Both habituation and sensitization are made possible due to changes in the strength of synaptic transmission between sensory and motor neurons. Synapses help us to store memories. Let's talk about habituation at the synaptic level. If you want to read more about this, I actually posted this link from this paper that was published by Kandel and his team back in 1978. So this is some really old research, but it was pivotal for the foundation of the field of neuroscience. But in this image, we're going to be able to walk through the different things that are occurring during habituation. So first, we know that repeated action potentials from repeated siphon stimulation could lead to diminished quantity of calcium in the terminal. Now remember, calcium is super important during this exocytosis, the process in which these neurotransmitter-containing synaptic vesicles are brought down to the synaptic cleft where they will release neurotransmitters into the synaptic cleft. Usually, especially during the very first siphon stimulation, there is going to be a large release of calcium in response to this. However, Kandel and colleagues found that repeated stimulation of the siphon induced habituation, thus inducing diminished calcium in the terminal. There's less vesicle docking here, less neurotransmitter release, and thus this results in habituation to the water jet. Sensitization is another form of non-associative learning that's been thoroughly studied in the Aplasia californica. Remember, sensitization is a form of learning that intensifies your response to all stimuli. Eric Kandel and his colleagues utilized a strong stimulus, which was a tail shock, to increase the gill withdrawal reflex response. And they found that this occurred via multiple neural pathways. We're going to walk through sensitization in the next slide. First, a tail shock activates serotonergic neurons in the tail. So as you can see, this sensitizing stimulus would activate serotonin-producing neurons. Serotonergic neurons in the tail then release serotonin at the synapse. Serotonin then binds the receptors on the sensory neuron. Next, serotonin stimulates a rise in cyclic adenosine monophosphate, known as CAMP. Remember, this is a second messenger that regulates gene transcription. Next, CAMP activates protein kinase A, known as PKA, in the sensory nerve terminal. Ultimately, PKA facilitates glutamate release when the siphon is touched. My key takeaway here is that glutamate is the main excitatory neurotransmitter that's going to be involved in EPSPs. And glutamate release temporarily strengthens the connection between the sensory and the motor neuron. How does long-term learning occur? Well, long-term sensitization requires both protein synthesis and new gene expression. Thus, we'll go back to the central dogma of biology: transcription of DNA to mRNA and translation of mRNA to protein. Remember that this process strengthens the synapses and increases learning. Thus, long-term learning or long-term sensitization can actually induce changes in gene expression. And if you want to read more about this, I've included some primary literature below. Let's walk through the steps of long-term sensitization. First, active protein kinase A goes to the nucleus. Remember that pKa actually helps to regulate sugar and lipid metabolism in the cell. Second, pKa activates the transcription factor CREB. This induces transcription of DNA to mRNA. Then mRNA is translated into protein outside of the nucleus, and new proteins can stimulate synapse growth. Ultimately, the creation of protein is crucial for learning to occur. There are two key types of associative learning. You've probably heard of them in your other psychology classes. This includes classical conditioning and operant conditioning, which is also known as instrumental conditioning. Classical conditioning was discovered by Dr. Ivan Pavlov. He looked at how conditioned stimuli and unconditioned stimuli paired together could motivate behavior, which we're going to talk about more on the next slide. Also, operant conditioning was discovered by Edward Thorndike, and reward and punishment that motivate behavior was studied thoroughly by B.F. Skinner. First, let's talk about classical conditioning, and I'm going to use the classic example of Ivan Pavlov's dog. But before we get into that experiment, let's go over key definitions. Remember that an unconditioned stimulus evokes a response without prior learning. This means that it typically has an innate meaning to the organism. So, for example, a dog is going to have a response to meat without prior learning. An unconditioned response occurs before learning. So a dog will likely salivate at the presence of food such as red meat, and that is innate within them. A conditioned stimulus originally elicits no response. So for example here, a bell would not typically generate a response in a dog. A bell is usually not very exciting to a dog. However, a conditioned response is when that bell is paired with an unconditioned stimulus like meat, which then evokes a conditioned response. Repeated pairings of a conditioned stimulus and unconditioned stimulus, in the case of this experiment, a bell and meat, evoked a conditioned response of salivating in response to hearing the bell. Let's walk through this experiment in a little more detail because I want to make sure that this is very clear to everyone. So first, before conditioning, when the dog heard the conditioned stimulus, a bell, it originally did not have a response. However, meat is an unconditioned stimulus that is going to induce salivating in the dog. When the sound of the bell was paired with presentation of red meat, this repeated pairing induced a conditioned response, in which the dog would now respond to the sound of a bell by salivating because it anticipated the reward of food. This is a classic example of classical conditioning. Now next we're going to talk about classical conditioning in the context of the Aplasia californica. Now let's utilize an example of classical conditioning in Aplasia californica. Again, this is some of our pivotal research in neuroscience, and Eric Kandel and his colleagues published this research back in the 80s. So first, in order to study classical conditioning, they needed to establish a conditioned stimulus and an unconditioned stimulus. The conditioned stimulus was water jet stimulation on the siphon, and the unconditioned stimulus was a tail shock because innately a slug does not want to receive that aversive shock. During the training phase, the CS and the US were paired. Thus, the slug would receive water jet stimulation on the siphon with a tail shock. This elicited a strong gill withdrawal response. Interestingly, during the testing phase, the CS, the water jet stimulation on the siphon, was presented alone, without the tail shock present. Without the tail shock, this still elicited a strong withdrawal response of the gill because it had previously been paired with the shock. So just stimulation of the water jet alone was enough to induce this conditioned response and this withdrawal response of gills. This learned withdrawal response increases neurotransmitter release to induce long-term learning. Now let's talk about the biological mechanisms underlying classical conditioning in aplasia californica. Now we've already talked about how repeated pairings of the water jet and the shock induced a conditioned response in which just the water jet alone could induce gill withdrawal because the sea slug had learned that a shock was previously paired with the water jet. So these repeated pairings actually induce calcium release from the presynaptic sensory neuron. Now this goes back to this first example I talked about. Remember, sensory neurons that innervate their skin are going to synapse onto motor neurons that innervate their muscle. And so thus this communication between the sensory neuron and the motor neuron can induce this gill withdrawal reflex. Now, during this time, when calcium is released, remember calcium performs exocytosis, this release of neurotransmitters contained in synaptic vesicles. When synaptic vesicles are carried to the presynaptic terminal, neurotransmitters can be dumped into the synaptic cleft. So calcium is playing an important role here in classical conditioning. It increases adenyl cyclase activity. Now, adenyl cyclase, remember, converts adenosine triphosphate, known as ATP, the cell's main energy source, to cyclic adenosine monophosphate, or CAMP. This is a key second messenger that regulates sugar and lipid metabolism, as well as cell growth and differentiation. Ultimately, CAMP increases pKa, which in turn phosphorylates protein and induces conformational change. This can induce new protein synthesis and increase the amount of glutamate that can be released by the sensory neuron onto the motor neuron. We know that glutamate is the main excitatory neurotransmitter, and thus this could generate excitatory postsynaptic potentials in the postsynaptic motor neuron. Simultaneous EPSPs together can form another action potential if a stimulus is very strong or salient. So these are the biological mechanisms underlying classical conditioning in the aplasia californica. Next, we're going to talk about a topic called long-term potentiation. This is essentially a way that synapses can be bolstered and strengthened. Long-term potentiation, also known as LTP, means to strengthen or facilitate or bolster. And LTP specifically is the strengthening of synapses. So they are patterns of strengthening and weakening that can lead to refined learning and memory. If you remember, we've talked about how synapses can typically be strengthened during different stages of sleep. For example, we've talked about how synapses can be strengthened during REM sleep. Well, LTP is the main form of synaptic plasticity, and researchers infer that this is a cellular mechanism behind learning and memory, especially long-term declarative memories. The NMDA receptor plays a key role in long-term potentiation. Now, if you remember, the NMDA receptor is the main glutamate receptor in the brain. It's both ligand and voltage gated, so it relies on its friend, the AMPA receptor, to generate depolarization in the postsynaptic cell. Because remember, the NMDA receptor is not going to be permeable to ions unless the cell itself becomes depolarized and glutamate has bind to it. Magnesium ions typically block this pore, and it isn't until the cell becomes more positively charged that magnesium can be removed to allow ions into the postsynaptic neuron. Now, when a glutamatergic synapse forms, the NMDA receptor is the only one in the postsynaptic membrane. The AMPA receptors appear after a while, and this is odd because it takes so much more time for them to fire. When there's strong activation of NMDA, it causes great strengthening of synaptic transmission. And this is what we're referring to, long-term potentiation, or LTP. Now, if you remember, The NMDA receptor is different from the AMPA receptor in another way. The NMDA receptor is also permeable to calcium, and this is what's going to induce the strengthening of synaptic transmission called long-term potentiation. So let's take a closer look at long-term potentiation. So as you can see in these images, the NMDA receptors are red and the AMPA receptors are blue. And we can see in all of these examples, there are 3 presynaptic neurons and 3 postsynaptic neurons. We're going to look at each stage of long-term potentiation. Now, before long-term potentiation, we don't see any activity. But during long-term potentiation induction, we can see that calcium is being released and allowing the release of neurotransmitters from these synaptic vesicles. Essentially, these neurotransmitters are going to come and bind to either the AMPA or NMDA receptors. For example, glutamate can come and bind to the NMDA receptor, which is going to allow an influx of calcium into the postsynaptic neuron, because we know that the NMDA receptor is specifically permeable to calcium, although the AMPA receptor is not. So after LTP occurs, this is going to induce these long-term changes. And calcium flooding into the postsynaptic neuron is what's playing a key role in long-term potentiation. This increase in synapse effectiveness can last a lifetime. Long-term potentiation leads to long-term memories. So as we can see here from our textbook, long-term potentiation can last a very long time. In this particular experiment, which we'll refer to this graph in a little bit, LTP was induced with stimulation using electrodes implanted in the hippocampus of an awake rat. And each data point shown in red here is the amplitude of the EPSP evoked with electrical stimulation. And so, interestingly, we can see that LTP was still evident a year later within the study. So the x-axis here is showing us the amount of days, whereas the y-axis is showing us the EPSP, or excitatory postsynaptic potential amplitude. So of course, on day 0 of this experiment, in which the rat's hippocampus was stimulated with electrodes that were implanted, we see this high frequency stimulation. And interestingly, these long-term postsynaptic changes induce long-term memory. As you can see, LTP was still evident a year later due to the generation of excitatory postsynaptic potentials that were occurring simultaneously. So let's quickly review what we've learned so far. Which brain area is crucial for procedural memory? Remember, we talked about how the striatum and the basal ganglia played an important role in these types of memories. What are 2 key types of associative learning? We've talked about habituation and sensitization. Why are Aplasia californica useful subjects to study learning and memory? Remember, they have extremely large nerve cells that can be recorded via microelectrodes. After aplasia have habituated to repeated water jet stimulation, what happens to the postsynaptic motor neuron? Remember that the postsynaptic motor neuron is going to have a reduced response or reduced gill withdrawal due to this habituation of the water jet. And there's also reduced neurotransmitter release from the presynaptic sensory neuron, leading to reduced EPSPs in the motor neuron, thus reducing this withdrawal reflex. What molecular process is crucial for long-term learning to occur? Remember, this is known as a long-term potentiation. Now we're going to focus on declarative memories, which are also known as explicit memories. Now there are 2 key types of declarative memories, including semantic memory and episodic memory. Now remember, semantic memories can be facts, such as geography, memorizing where different states or countries are located. And episodic memories are events such as your wedding day. These are largely regulated in the medial temporal lobe. If you remember, the hippocampus sits in the medial temporal lobe as well as the amygdala, and the diencephalon, which is known as the thalamus in the fully grown adult brain. Although we've talked about different types of memories, I want to note that memories can also be classified by time. So, for example, if you take in new sensory information, your working memory and your short-term memory are going to process that information. And throughout time, the consolidation of these memories can then turn into long-term memories. Remember that long-term potentiation plays an important role in this consolidation of a long-term memory. Now, encoding, consolidation, and retrieval of long-term memories is what we're going to focus on for the rest of this lecture. We're going to talk about 3 key ways that memories can be classified by time. The first is a long-term memory. So long-term memories are memories that last months or years. So not all memories retain, right? Long-term memories could allow us to remember a classmate that we had perhaps in grade school. When you look at a yearbook like this, you could look at old pictures and recognize someone from your childhood. Short-term memory lasts seconds to hours, and they're very vulnerable to disruption. We know that things like synaptic pruning can also prune away those synapses if they are not going to then be consolidated into long-term memories. Working memory is also very interesting because it has a limited capacity. It requires a lot of rehearsal. So, for example, a phone number, memorizing a phone number is a great way to think about working memory. What brain areas are involved in declarative memory? I'm sure you guessed it. Of course, we're looking at the limbic lobe again. So how does information flow through the medial temporal lobe? Now remember, whenever we've looked at the medial temporal lobe, I always talk about the hippocampus, which sits right here. This is a curved structure, so the more dorsal part of the hippocampus is up here. And we've talked about many of these areas of the brain, like the fornix and the cingulate cortex and the hypothalamus, that all play an important role in the loading of information and processing those memories. So when we take in sensory information, cortical areas like the cingulate cortex can process those. And during this time, they could be like a short-term memory. Now, the parahippocampal areas are going to take in this information. It's going to be sent to the hippocampus and then to the thalamus and hypothalamus via the fornix. Now, remember, the fornix is a commissure. This is a large bundle of axons that can connect structures between the brain. And so the fornix specifically helps with communication between the hippocampus and the thalamus and the hypothalamus. So there's evidence for the importance of the prefrontal cortex in working memory. And a great example of this, we're going to go back to our friend Phineas Gage. If you remember, Phineas Gage had a terrible accident on the railroad where this large iron rod went through his prefrontal cortex. It entered from the bottom of his eye and went through his entire brain and skull. Ultimately, after this accident, Phineas Gage had a lot of different behavioral and physiological symptoms. For example, he had changes in personality. It was hard for him to plan and stay organized. It affected his work. Ultimately, we now know that the prefrontal cortex plays a really important role in decision-making, morality, personality, and thus Phineas Gage had difficulty planning and organizing behaviors to complete a task. Thus, we now understand that the prefrontal cortex plays an important role in our working memory. Remember, an example of working memory is memorizing and learning a phone number. And of course, brain areas that are important for declarative memory, so these could be semantic or episodic memories like facts or events, include the medial temporal lobe and the thalamus. So, again, these are the types of memories like geography or your wedding day, these memories that you need to store long term. That doesn't necessarily mean that these memories are going to be stored in the medial temporal lobe and thalamus. They can be stored in other areas like the cingulate cortex. But ultimately, the hippocampus serves as a loading dock for memories. If you remember, the hippocampus plays a really important role in loading that new information. A great example of this is with patient HM. If you remember, patient HM had severe seizures, so in an experimental surgery, they removed his entire hippocampus. After the surgery, he could remember his childhood memories and long term memories, but it was difficult for him to create new memories. And that's because the hippocampus helps to facilitate these new memories. That's why I refer to it as this loading dock for declarative memory. Interestingly, if you actually stimulate the temporal lobe, people have random episodic flashbacks of memories from their past. And again, just because you stimulate this area and you have these flashbacks of your memories, it doesn't necessarily mean the memories are stored there, but it does show that the hippocampus plays an important role in both consolidation of memories and that reactivation and reconsolidation of memories you want to recall back into your working memory. So here's the mouse hippocampus in 3D, and I thought I would just share this with you guys. It's a really cool tool. This was developed by USC and their Neuroimaging Institute, and you can see and watch this mouse hippocampus in 3D. And you can actually go onto their website, which I've attached a link for this, and you can look at all of the different components of the mouse hippocampus, which is pretty cool if you're nerdy like me. Now let's talk about the simple circuits of the hippocampus. So we've only talked about a few key aspects of the hippocampus. We haven't actually looked at the circuits within the hippocampus. And we're going to look at this in a mouse brain specifically because they're a great model for the human brain. So we're going to start over here at step 1. So essentially when we take in information, it's going to flow from this area called the entorhinal cortex to the dentate gyrus, shown here in step 2. The dentate gyrus is an amazing area of the hippocampus where neurogenesis can occur even in adulthood. And we're going to talk more about neurogenesis in adulthood in a few slides. The dentate gyrus then sends information to the CA3 region, shown here, via these mossy fiber synapses. These are really just axons that are communicating and synapsing between the dentate gyrus and the CA3 region. And then these CA3 cells are going to give rise to axons that can leave the hippocampus. So these are longer axons that can branch into the fornix. And if you remember, the fornix is this commissure that allows for communication between the hippocampus and the thalamus and the hypothalamus, because axons from the hippocampus can project to these other areas. So ultimately, the fornix is this main mode of communication, and information can thus be sent from the hippocampus to the thalamus and the hypothalamus. Again, I always like including tools for you guys. This is awesome. I provided a link to this website from USC again. This is a reference atlas of the mouse hippocampus, and you can look at all the different areas of the mouse brain specifically. So as you can see, I was kind of playing around with this earlier. We're looking at a coronal section of the mouse brain, and we can actually highlight and look at different circuits within the brain. So I was looking at the dentate gyrus specifically in the hippocampus, shown here, the DG. And this tool allows you to look at any of the different circuits of the brain you're interested in. So here's our hippocampus right here. Now let's talk about neurogenesis, which is the creation of new neurons in the adult brain. So luckily, we do create new neurons throughout our life. And we're going to talk about different ways that where you can increase the production of new neurons and which areas of the brain are likely going to have neurogenesis occurring. Interestingly, we weren't able to study neurogenesis in much detail until after World War II. And so I'm going to give you some backstory about this. Between the years of 1955 and 1963, we know that there were hundreds of nuclear bombs that were detonated in atmospheric tests, as shown here in Figure A. This caused widespread decimation of radioactive fallout, and so during this time, as we can see in this graph, there was a large spike in this radioactive isotope called carbon-14C. And 14C was incorporated in all living things, including human DNA, specifically including the replicating DNA of human neurons. So essentially, this put a timestamp on every cell born during this time. And this allowed scientists to measure new neurons that had developed after this timestamp. This study was conducted by Spalding and colleagues, and they found that 700 new neurons are added to the hippocampus in the adult brain every single day. And so, interestingly, it wasn't until this timestamp from the incorporation of radioactive isotope carbon-14C that we could fully understand neurogenesis and how many new neurons occurred. So this again is one of those happy accidents that led to increased knowledge in the field of neuroscience. So what increases neurogenesis? You've probably heard me talk about this a lot in class before, but exercise is a great way to enhance neurogenesis. Rats that are given an exercise wheel and have the ability to run on it daily have increased levels of neurogenesis. Exercise has been shown to increase an important neurotrophic factor called brain-derived neurotrophic factor, BDNF, which I've talked about a lot in class, but essentially BDNF increases neurogenesis in the hippocampus, and thus high levels of BDNF increase the number of neurons and correlate with enhanced performance on memory tasks that require the hippocampus. So ultimately, exercise is one of the best ways to increase the production of new neurons. There are two key sites for neurogenesis in the adult brain, including the olfactory bulb shown here and the dentate gyrus shown here. We already talked about how the dentate gyrus is a part of the hippocampus. So although we can create new neurons in these two areas, it's sometimes hard for the regrowth of CNS axons to occur. So why can't we harness this ability for regrowth of CNS axons after damage? Well, there are a few different reasons. First, there's something called glial scars, in which pro-inflammatory cytokines that chronically activate glial cells leave scars that prevent the regrowth of new cells. There are types of glial cells in the peripheral nervous system, like Schwann cells, that can actually regenerate axons after injury, but oligodendrocytes, which are essentially Schwann cells within the central nervous system, cannot do so. This is due to a factor called no-go. This is a glial factor that inhibits axon growth released when oligodendrocytes are damaged. How did we discover the key functions of the hippocampus? Patient HM is a primary example of our understanding of the hippocampus. So remember, patient HM was suffering from seizures, and in an experimental procedure, they removed his entire hippocampus, as shown here. So this is patient HM's brain next to a normal, healthy brain. Ultimately, after the removal of his hippocampus, his working and procedural memory remained intact. But this experiment led us to understand the medial temporal lobe is critical for memory consolidation, but quite as much for the retrieval of memories. Now, there are two different forms of amnesia that can develop from brain trauma. Patient HM developed something called anterograde amnesia, which I'm going to touch on in a moment. But there are two different types I'm going to talk about, including retrograde and anterograde. Retrograde amnesia, as shown here, is when a person has difficulty retaining memory prior to their accident or time of trauma, but can still develop new memories. This is different from anterograde amnesia. This is what patient HM experienced. He could remember things like procedural memory and working memory. However, after his hippocampus was removed, he had difficulty creating new memories. So we know the hippocampus helps facilitate this. Again, it's kind of a loading dock for memories. So ultimately, anterograde amnesia resulted in his inability to create new memories. These different types of amnesia have been thoroughly utilized in Hollywood in movies like 50 First Dates or The Vow. If you've seen the movie 50 First Dates, this is a great movie with Drew Barrymore and Adam Sandler. Drew Barrymore gets in a terrible car accident in which she has a traumatic brain injury, and she develops anterograde amnesia, in which she can no longer develop memories after her accident. She only has memories intact from the day before her accident and her trauma. So it's pretty interesting how Hollywood has also utilized some of these neuroscience topics. There are three key functions to the hippocampus, to put in very simple terms. The first is binding sensory information and consolidating memories. So I love this example here. The hippocampus is essentially taking in information and sending that to the cortex for long-term storage. So it does help with long-term memory storage during consolidation. Also plays a very important role during spatial memory, which we're going to touch on next. Spatial memory allows us to remember the location of places. This helps us with spatial navigation. And this phenomenon has been studied a lot in London taxi drivers. Now, you might think this is kind of funny, but if you've ever been to London, you know that it's very busy. There are taxis everywhere, and it's really hard to navigate the city. And London taxi drivers have to take a very difficult test in which they have to memorize all of the streets of London to get their license. So, researchers have actually looked at the relationship between the size of the hippocampus and experience in being a London taxi driver. And they found that London taxi drivers actually have a larger posterior hippocampus than control subjects. Thus, the posterior hippocampus is associated with helping us with our cognitive mapping skills, our spatial navigation. So this was super interesting to find. Also, in PET imaging studies, as shown here in your textbook, this was looking at the activity in the brain related to spatial navigation. And participants were given this virtual town to look at. It was shown on a computer monitor. And subjects in a PET imaging machine used buttons to navigate this virtual environment. And they found that when they were trying to learn this new virtual town, they had increased brain activity associated with spatial navigation. And this was occurring right in the hippocampus and left of the caudate nucleus, specifically as shown here in yellow. So how can we measure learning and memory in rodents? So here's my first example of a behavioral paradigm that's utilized in animal research to measure learning and memory. This is called the radial arm maze. This is a maze with arms that radiate from a central point, and rats learn to navigate it to find rewards at the end of the arms. So this measures learning and memory by testing the rat's ability to recall arm locations and the rewards where they're placed. This assesses working and reference memory, as well as identifying errors like revisiting arms without rewards. So researchers use this to study brain mechanisms underlying learning and memory. The Morris-Water maze is another behavioral paradigm that's utilized to study learning and memory in rodents. So as you can see here, the Morris water maze is a circular pool and it's filled with opaque colored water. So rats have to learn to locate a hidden platform shown here just beneath the water surface utilizing different spatial cues. So this test measures learning and memory by testing rats' ability to find this platform and assesses spatial memory and learning strategies. So researchers typically use this to study brain mechanisms underlying spatial navigation and memory formation. And what's also interesting to note here is that both long-term potentiation and spatial learning require NMDA receptor activation. Another behavioral paradigm utilized to measure spatial memory impairment is the object location placement test. And so in this task, mice or rats are given the opportunity to explore two identical objects in an arena during this training phase. But during testing, about 24 hours later, the positions of the objects are changed. As you can see, this object moved to the opposite corner. So it's different from its original arrangement. And this change challenges the rats or mice to recall the initial object locations. If mice spend more time with the object that was moved to the new location, this is indicative of better spatial memory. So ultimately, rodents learn the object locations and later need to be able to remember this original arrangement in order to indicate that they have better spatial memory. So this measures spatial memory and the ability to recall object locations, and it provides insights into memory processes and brain function that's related specifically to spatial cognition.